Dr. Sarah Chen had been teaching molecular biology at Stanford University for 12 years, but she had never encountered a student quite like Marcus Rodriguez. While other students in her advanced human genetics course scribbled notes and nodded along during lectures, Marcus always sat in the third row, left side, with his laptop open and a questioning expression that made Sarah both nervous and excited. On that particular Tuesday morning in September, Sarah was explaining the basics of human genetic composition to her class of 38 graduate students. The lecture hall was filled with the usual sounds of typing keyboards and occasional coughs as she clicked through her presentation slides. Human DNA consists of approximately 3.2 billion base pairs, Sarah explained, pointing to the colorful diagram on the large screen behind her. We share about 99.9% of our DNA with each other, which is why we're all recognizably human despite our differences in appearance, personality, and abilities. She moved to the next slide, showing a pie chart of genetic composition. About 2% of our genome consists of protein-coding genes, which are the instructions for making the proteins our bodies need to function. Another 24% consists of introns, which are segments within genes that don't code for proteins but may have regulatory functions. Marcus raised his hand, as he often did. Sarah nodded toward him, expecting another of his thoughtful questions about genetic expression or evolutionary biology. Professor Chen, Marcus said, his voice carrying a hint of confusion that was unusual for him. Your chart shows that we understand the function of about 92% of human DNA. What about the other 8%? The chart just labels it as other sequences, but what does that actually mean? Sarah paused, her marker hovering over the whiteboard. It was a fair question, one that she typically glossed over because the answer was somewhat embarrassing for the scientific community. She looked at the faces staring back at her, noting that Marcus's question had captured the attention of several other students who had been half listening. That's an excellent question, Marcus, Sarah said, buying herself a moment to think. The remaining 8% consists of various sequences that we haven't fully characterized yet. Some of it is likely regulatory DNA that we simply don't understand, some might be evolutionary remnants, and some could be. She trailed off, realizing that her usual vague explanation wasn't going to satisfy Marcus or the other students who were now leaning forward with interest. Marcus frowned and opened his laptop, his fingers flying across the keyboard. Professor, I've been looking at the latest genomic databases during your lecture. The Human Genome Project was completed over 20 years ago, and we've had massive advances in sequencing technology since then. How is it possible that we still don't know what 8% of our own genetic code does? The question hung in the air like a challenge. Sarah felt a familiar flutter of excitement mixed with apprehension. Marcus had a talent for asking the questions that kept her awake at night, the ones that made her wonder if she was teaching outdated information or if there were gaps in human knowledge that deserved more attention. You're right to be curious about this, Sarah said, setting down her marker and facing the class directly. The truth is that 8% represents one of the biggest mysteries in human genetics. We call some of it junk DNA, though many scientists now believe the term is misleading and potentially harmful to our understanding. A student named Jennifer raised her hand. But, Professor, if it's been over 20 years since the Human Genome Project, why haven't we figured it out? We can edit genes with CRISPR, we can sequence entire genomes in hours. Surely we can identify what a few million base pairs are doing? Sarah walked over to her desk and pulled out a thick folder filled with research papers. Jennifer, that's exactly what makes this so fascinating and frustrating. We have the technology to read the genetic code perfectly. We can see every single nucleotide. But reading the code and understanding what it means are two completely different things. She opened the folder and pulled out a recent paper from Nature Genetics. This study from last year attempted to characterize some of these unknown sequences. The researchers found that when they tried to delete small sections of this mysterious 8% in laboratory mice, some of the mice developed unusual traits. Marcus leaned forward. What kind of unusual traits? Enhanced cognitive abilities in some cases, unusual stress responses in others, and some developed what the researchers described as anomalous behavioral patterns that didn't fit any known genetic syndrome, Sarah explained, reading directly from the paper's abstract. The classroom had grown completely quiet. Even the students who usually spent lectures checking their phones were now paying attention. But, here's where it gets really interesting, Sarah continued. When the researchers tried to replicate their experiments, they couldn't get consistent results. Some mice showed the enhanced traits, others showed no change at all, and a few showed completely different traits that hadn't appeared in the original experiment. A student named David raised his hand. That sounds like the DNA might have different functions in different individuals. Is that possible? Sarah nodded slowly. It's possible, but it would be unprecedented. Most genetic sequences have consistent functions across individuals and even across species. The fact that these sequences seem to behave differently in different subjects suggests that we might be dealing with something we don't yet understand about how DNA works. Marcus was typing furiously on his laptop again. After a few moments, he looked up with an expression that Sarah had learned to recognize. It was the same expression he'd worn when he'd asked her about the mathematical inconsistencies in Hardy-Weinberg equilibrium calculations, leading to a semester-long independent study project. Professor Chen, Marcus said carefully, I've been cross-referencing the papers you mentioned with some recent studies on human genetic variation. There's something strange about the distribution of this unknown 8%. What do you mean? Sarah asked, though she suspected she knew where this was heading. It's not randomly distributed across the human population, Marcus explained, turning his laptop screen toward the class. People of certain geographic ancestries have slightly different compositions within that 8%. Not different amounts, but different sequences. And, the differences don't match any known migration patterns or evolutionary timelines. Sarah felt her pulse quicken. She had noticed this pattern in her own research but had assumed it was simply a reflection of normal genetic diversity. The fact that Marcus had identified it independently suggested that the pattern was more significant than she had realized. Can you be more specific about the geographic patterns? Sarah asked, moving closer to see Marcus's laptop screen. People whose ancestors came from certain regions in Central Asia, parts of the Amazon basin, and some isolated Pacific islands all share similar sequences within this 8%, Marcus explained. But, these populations had no contact with each other for thousands of years, if ever. There's no evolutionary reason why they should share genetic material that other human populations don't have. The implications of what Marcus was suggesting began to dawn on Sarah and several of the other students. If populations with no historical contact shared unique genetic sequences, it suggested either that the sequences were much older than previously thought, or that they had been acquired through some mechanism other than normal inheritance. Jennifer raised her hand again. Professor, could this 8% be viral DNA that got incorporated into the human genome? I know that happens sometimes. That was one of the first theories researchers investigated, Sarah replied. We do carry ancient viral DNA in our genomes, but most of it is degraded and clearly identifiable as viral in origin. This 8% doesn't match any known viral sequences, and it appears to be actively maintained rather than degrading over time. Marcus looked up from his laptop with a troubled expression. Professor, there's something else. I've been looking at recent medical literature, and there are case reports of people who seem to have unusual variants within this 8%. Some of them report enhanced memory, unusual sensory sensitivity, or other traits that don't fit normal human variation. Sarah felt a chill run down her spine. She had seen similar reports but had dismissed them as anecdotal or the result of other genetic factors. The fact that Marcus was finding the same patterns independently made her reconsider. Marcus, I want you to be very careful about drawing conclusions from medical case reports, Sarah warned. There are many factors that could explain unusual traits, and genetic correlation doesn't necessarily imply causation. I understand that, Professor, Marcus replied. But, the pattern is consistent across hundreds of cases. People with specific variants in this unknown 8% consistently report similar unusual abilities or experiences. And, many of them come from the same geographic populations I mentioned earlier. The bell rang, signaling the end of class, but none of the students moved to leave. The questions Marcus had raised were too compelling to abandon for their next lectures. Sarah looked around the classroom at the expectant faces of her students. She had always prided herself on being honest about the limitations of scientific knowledge, but Marcus's questions were pushing into territory that made her genuinely uncomfortable.
"Here's what I want you all to do," Sarah said finally. "This is going to be our class project for the remainder of the semester. I want you to work in teams to investigate different aspects of this unknown 8%. Marcus, I want you to lead a team focusing on the geographic distribution patterns you've identified.
Jennifer, I want your team to investigate the viral DNA hypothesis more thoroughly. David, your team can focus on the medical case reports and see if there are any consistent patterns." She paused, looking directly at Marcus.
"But I want everyone to remember that we're scientists, not conspiracy theorists. We follow the data wherever it leads, but we don't jump to conclusions without evidence.
And we certainly don't make claims that could cause panic or promote pseudoscientific thinking."
Marcus nodded seriously. "Professor, I understand the responsibility. But if this 8% of human DNA is doing something that we don't understand, don't we have an obligation to figure out what it is? Especially if it might be affecting human health or abilities?"
Sarah gathered her papers and closed her laptop, considering Marcus's question.
"Yes, Marcus, we do have that obligation.
But we also have an obligation to approach this mystery with proper scientific rigor and skepticism." As the students finally began to file out of the classroom, Sarah remained behind, staring at the genetic composition chart still displayed on the screen. The colorful pie chart that she had shown hundreds of times suddenly seemed inadequate, even misleading. That innocent-looking slice labeled other sequences represented millions of base pairs of DNA that existed in every human cell, yet remained completely mysterious to science. For the first time in her teaching career, Sarah realized that she might be on the verge of uncovering something that would change not just her understanding of genetics, but potentially humanity's understanding of itself. The thought was both thrilling and terrifying. She pulled out her phone and scrolled through her contacts until she found the number for Dr. Elena Vasquez, her former colleague who now worked at the National Human Genome Research Institute. If anyone would have access to the latest research on uncharacterized human DNA sequences, it would be Elena. The phone rang 3 times before Elena answered. "Sarah. What a surprise.
How's the teaching life treating you?"
"Elena, I need to ask you about something," Sarah said without preamble.
"How much do we really know about the function of uncharacterized sequences in the human genome?"
"Specifically, I'm talking about sequences that make up about 8% of the genome but don't fit into any of our standard categories."
There was a long pause on the other end of the line. "Sarah, that's a very specific question. Is there a particular reason you're asking?"
"One of my students has been asking some very pointed questions about gaps in our knowledge, and I realized I don't have good answers," Sarah explained.
"I'm starting to wonder if we've been too quick to dismiss sequences we don't understand as unimportant. Can you meet me for coffee tomorrow?" Elena asked, her voice carrying a tone that Sarah couldn't quite identify. "There are some things we should discuss in person."
After hanging up, Sarah stood alone in her empty classroom, staring at the genetic chart on the screen. She had the distinct feeling that Marcus's simple question about the mysterious 8% was about to lead her down a path that would challenge everything she thought she knew about human biology.
The next 24 hours couldn't pass quickly enough.
Sarah arrived at the small coffee shop near the National Human Genome Research Institute 15 minutes early, her mind racing with questions about Elena's cryptic response to her inquiry about uncharacterized DNA sequences. She had barely slept the night before, alternating between excitement about the potential research project and anxiety about what Elena might tell her. Elena appeared promptly at 2 p.m., looking older than Sarah remembered but carrying herself with the same confident stride that had made her such an effective research leader during their graduate school days together. She carried a thick manila envelope under her arm and wore the kind of serious expression that Sarah had learned to associate with significant scientific developments. "Sarah, thank you for meeting me," Elena said, sliding into the booth across from her.
"Before we start talking, I need to ask you something. What prompted your student to ask about uncharacterized DNA sequences? Was it just academic curiosity, or had he been reading specific research?"
Sarah described Marcus's questions and his independent research findings about geographic distribution patterns. As she spoke, Elena's expression grew increasingly serious, and she occasionally glanced around the coffee shop as if ensuring they weren't being overheard. "Elena, you're making me nervous," Sarah said when she finished her explanation.
"What aren't you telling me about this 8%?
Elena opened the manila envelope and pulled out several research papers, most of which had been marked with red confidential stamps.
"Sarah, what I'm about to share with you is preliminary research that hasn't been published yet.
Some of it may never be published, depending on what we discover." She spread the papers across the table, revealing titles that made Sarah's pulse quicken: Anomalous Genetic Sequences in Human Chromosome 14, Unexpected Protein Expression from Non-Coding DNA Regions, and Geographic Clustering of Uncharacterized Human Genetic Variants. "Your student is more perceptive than he realizes," Elena continued. "The National Human Genome Research Institute has been quietly investigating these uncharacterized sequences for the past 3 years. We've been calling it Project Helix, and what we've found is challenging some fundamental assumptions about human genetics." Sarah picked up the first paper and began reading the abstract.
"Elena, this says that some of these sequences are actively transcribed into RNA and translated into proteins, but the proteins don't match any known human protein families." "That's correct," Elena confirmed.
"We've identified at least 47 distinct proteins that appear to be produced from sequences within that mysterious 8%. The proteins are functional, meaning they have specific shapes and appear to interact with other cellular components, but we have no idea what they do." Sarah looked up from the paper, her mind struggling to process the implications. "But that would mean that humans are producing proteins that we don't even know exist.
How is that possible? Wouldn't we have noticed the effects of these proteins in our physiology?" Elena pulled out a tablet and opened a file containing dozens of photographs. "That's where things get really interesting.
We think we are noticing the effects, but we've been attributing them to other causes." The photographs showed brain scans, blood analysis results, and other medical imaging. Elena scrolled through them as she explained, "We've been working with several major medical centers to identify people who have active variants of these uncharacterized sequences.
We call them expressors because their cells are actively expressing these unknown proteins." "What do you mean by active variants?"
Sarah asked, though she suspected she already knew the answer. "Not everyone has the same sequences within that 8%, Elena explained. Most people have what we call dormant versions, where the sequences are present but not actively transcribed. But about 3% of the human population has active versions that are producing these mystery proteins." Sarah felt a chill as she remembered Marcus's research about geographic distribution patterns.
"Elena, does this 3% correlate with specific populations or geographic regions?" Elena nodded grimly. "Your student was right about the geographic clustering.
People of Central Asian, Amazonian, and Pacific Islander ancestry are much more likely to be expressors.
But here's what's really strange, we're also finding expressors in populations where the ancestry doesn't fit the pattern."
She pulled out another paper and pointed to a data table. "We found expressors in urban populations in North America and Europe, people whose family histories show no connection to the regions where these sequences are common. And, in many cases, these people report that their unusual traits started manifesting recently, sometimes within the last few years."
"What kind of unusual traits?"
Sarah asked, though she remembered Marcus mentioning enhanced cognitive abilities and sensory sensitivity.
Elena looked around the coffee shop again before answering.
"Enhanced memory, improved pattern recognition, unusual stress resistance, and, in some cases, sensory abilities that appear to exceed normal human ranges. We had one subject who could detect electromagnetic fields at levels that shouldn't be perceptible to humans." Sarah set down the paper she had been reading, feeling overwhelmed.
"Elena, this sounds like science fiction. Are you telling me that some humans are developing superhuman abilities because of genetic sequences that we previously thought were junk?"
"I'm telling you that we're observing traits in certain individuals that correlate with the expression of unknown proteins from uncharacterized DNA sequences," Elena replied carefully.
"Whether those traits constitute superhuman abilities is a matter of interpretation."
She pulled out another document, this one bearing the seal of the Centers for Disease Control and Prevention.
"But, Sarah, there's something else." We're not the only ones who have noticed these patterns. The CDC has been tracking unusual medical reports that correlate with our expressor populations.
Sarah read the CDC document's summary, which described cases of individuals seeking medical attention for sensory overload, unexplained changes in sleep patterns, and altered responses to medications. Elena, some of these medical reports sound like people are having adverse reactions to expressing these proteins.
That's exactly what we think is happening, Elena confirmed.
It appears that when these sequences become active, they can cause significant physiological changes that the human body isn't necessarily adapted to handle. We're seeing cases of neurological stress, immune system irregularities, and metabolic disruptions. Sarah felt a growing sense of unease as she connected the dots.
Elena, if these genetic sequences are becoming active in people who previously had dormant versions, what's causing the activation?
Elena was quiet for a long moment, her fingers drumming nervously on the table.
That's the question that keeps us awake at night, Sarah.
We don't know what's triggering these activations, but they appear to be increasing in frequency.
She pulled out a graph showing activation rates over time.
3 years ago, when we started Project Helix, we estimated that about 1% of the population were expressors. Today, we estimate it's closer to 3%. If that trend continues, we could be looking at a significant portion of the human population expressing these unknown proteins within the next decade.
Sarah studied the graph, noting the sharp upward curve in recent years. Elena, this looks exponential. What happens if this continues? What happens if a large percentage of humans start expressing these proteins? We don't know, Elena admitted.
The proteins could be beneficial, helping humans adapt to environmental changes or providing new capabilities. Or they could be harmful, causing medical problems that we're not equipped to treat.
Or they could be something else entirely.
She paused, gathering the papers back into a stack.
Sarah, there's one more thing. We've found evidence that these sequences might not be entirely human in origin.
Sarah felt her world shift slightly.
What do you mean by that? Elena pulled out a final document, this one marked with security classifications that Sarah didn't recognize. Phylogenetic analysis of these sequences shows that they don't follow the same evolutionary patterns as the rest of the human genome.
They appear to have been inserted into human DNA at some point, but not through any natural process we understand.
Inserted by what? Sarah asked, though she wasn't sure she wanted to hear the answer. We don't know, Elena replied.
The sequences are too complex to be the result of random mutation, too organized to be viral in origin, and too recent to be explained by normal evolutionary processes.
They appear to have been deliberately designed and introduced into the human genome.
Sarah sat back in her chair, feeling as though the ground had shifted beneath her feet.
Elena, are you suggesting that someone or something has been modifying human DNA? Elena's expression was grave.
Sarah, I'm suggesting that we need to consider the possibility that the human genome contains elements that weren't put there by natural evolution.
And those elements are now becoming active at an unprecedented rate.
She closed the manila envelope and slid it across the table to Sarah. I want you to take these papers and review them carefully.
Then, I want you to talk to your student, Marcus.
Based on what you've told me about his research abilities and his questions, I think he might be exactly the kind of fresh perspective we need on this project. Sarah took the envelope, feeling its weight both literally and figuratively.
Elena, if what you're telling me is true, this changes everything we thought we knew about human evolution and genetics.
It changes more than that, Elena replied.
If these activations continue to increase, it might change what it means to be human.
As they prepared to leave the coffee shop, Elena placed a hand on Sarah's shoulder. Sarah, I need you to be careful about how you handle this information. There are aspects of Project Helix that involve national security concerns, and there are people who would prefer that this research remain classified indefinitely. Sarah felt a new wave of anxiety wash over her.
Elena, are you telling me that this research is being suppressed?
I'm telling you that there are legitimate concerns about how the public might react to learning that human DNA contains sequences of unknown origin that are causing physiological changes in a growing percentage of the population, Elena explained.
But I'm also telling you that we need scientists like you and your student working on this problem, because the official research efforts may not be moving fast enough.
After Elena left, Sarah sat alone in the coffee shop for another hour, staring at the manila envelope and trying to process everything she had learned. Her simple genetics lecture had somehow led her into a mystery that challenged fundamental assumptions about human biology and evolution. She thought about Marcus and his insightful questions about the mysterious 8% of human DNA. What had started as academic curiosity was now revealing itself to be one of the most significant scientific discoveries in human history, and potentially one of the most dangerous. Sarah pulled out her phone and scrolled to Marcus's contact information. She hesitated for a moment, knowing that involving him in Project Helix would change his life just as dramatically as it was changing hers. But Elena was right, they needed fresh perspectives and brilliant minds working on this problem. She typed a simple text message, "Marcus, can you meet me in my office tomorrow morning? I have some information about that 8% that you're going to find very interesting." After sending the message, Sarah gathered the envelope and headed back to her car. As she drove back to campus, she couldn't shake the feeling that she was standing at the threshold of a discovery that would fundamentally alter humanity's understanding of itself. The question that haunted her was whether that discovery would prove to be humanity's salvation or its doom. Marcus arrived at Sarah's office at precisely 8 a.m., carrying his usual laptop bag and a large coffee that suggested he had been awake for several hours already. Sarah had spent most of the night reviewing Elena's papers, and the dark circles under her eyes probably matched his. "Professor Chen, your text message was intriguing," Marcus said as he settled into the chair across from her desk. "I've been doing more research on those geographic distribution patterns, and I've found some things that I think you'll find interesting." Sarah set down her own coffee and pulled Elena's manila envelope from her desk drawer. "Marcus, before you share your findings, I need to show you something. But first, I need you to understand that what I'm about to tell you is not public information, and it carries serious implications for your future research and possibly your safety." Marcus's expression grew serious. "Professor, are you talking about classified research?" "I'm talking about research that reveals fundamental questions about human evolution and genetics that some people would prefer remained unanswered," Sarah replied. She spread Elena's papers across her desk, watching Marcus's eyes widen as he read the titles and classification markings. For the next hour, Sarah walked Marcus through Project Helix, explaining the discovery of unknown proteins, the identification of expressor populations, and the evidence suggesting that the mysterious 8% of human DNA might not be entirely natural in origin. Marcus listened intently, occasionally asking questions that demonstrated his growing understanding of the implications. "Professor, this explains something I discovered last night," Marcus said when Sarah finished her explanation. He opened his laptop and pulled up a complex data visualization. "I've been analyzing public health records for correlations with the geographic patterns we identified, and I found some disturbing trends." The visualization showed a map of the United States with colored dots representing various medical conditions. Marcus zoomed in on specific regions as he explained his findings. "I found higher than average rates of neurological disorders, autoimmune conditions, and sensory processing disorders in areas with higher concentrations of the populations we identified as having unusual variance in that 8%, Marcus explained. "But here's what's really interesting, the rates have been increasing significantly over the past 5 years." Sarah studied the data, recognizing patterns that aligned with Elena's information about increasing activation rates. "Marcus, this suggests that people are experiencing medical problems as these genetic sequences become active." "That's exactly what I think is happening," Marcus confirmed. "But, Professor, there's something else. I also found increased rates of exceptional performance in standardized testing, enhanced academic achievement, and reports of unusual talents in the same populations." He switched to another visualization showing educational and cognitive performance metrics. "It's as if the activation of these genetic sequences is having both positive and negative effects, sometimes in the same individuals." Sarah felt a chill as she considered the implications. "Marcus, if these activations are causing both enhanced abilities and medical problems, we might be looking at an evolutionary transition that the human body isn't adapted to handle." Marcus nodded grimly. Professor, there's one more thing. I've been tracking social media posts and online forums where people discuss unusual experiences or abilities. I've found thousands of posts from people describing symptoms that match what you've shown me about expressor populations. He opened a folder containing screenshots from various social media platforms. Sarah read through posts describing enhanced sensory perception, unusual memory abilities, electromagnetic sensitivity, and various neurological symptoms. Marcus, this suggests that there might be far more expressors than the official research estimates, Sarah observed. If people are experiencing these changes but not seeking medical attention, the actual numbers could be much higher.
That's what I think too, Marcus agreed. Professor, I've been corresponding with some of these people online, using anonymous accounts.
Many of them report that their unusual experiences started within the last 2 to 3 years. Sarah leaned back in her chair, feeling overwhelmed by the scope of what they were discovering. Marcus, if the activation rate is accelerating and affecting more people than we realize, we need to understand what's triggering these activations.
Marcus pulled up another document on his laptop. Professor, I think I might have found a clue about that.
I've been analyzing environmental data for correlations with activation hotspots, and I found something strange. The document showed maps overlaying activation rates with various environmental factors including electromagnetic field strength, chemical pollutant levels, and radiation exposure.
Marcus pointed to specific regions where multiple factors seemed to correlate.
Areas with higher levels of electromagnetic radiation from cell towers, power lines, and wireless networks show increased activation rates, Marcus explained.
But it's not just electromagnetic fields. There are also correlations with certain chemical pollutants and even cosmic radiation levels.
Sarah studied the data, noting that the correlations were strong but not absolute.
Marcus, this suggests that environmental factors might be triggering the activation of these genetic sequences. But, if that's true, then modern technology and environmental changes could be causing widespread genetic modifications in humans.
Professor, that's exactly what I think is happening, Marcus said. And if these environmental triggers continue to increase, we could be looking at a scenario where most of the human population experiences these genetic activations within our lifetime. Sarah stood up and walked to her office window, looking out at the campus where thousands of students went about their daily lives, unaware that their genetic code might be in the process of fundamental change. Marcus, we need to think carefully about what we do with this information, Sarah said. If we're right about environmental triggers causing genetic activations, the implications for public policy and human safety are enormous.
Marcus saved his files and closed his laptop. Professor, I've been thinking about that too.
But I keep coming back to the question of whether these genetic sequences were always present in human DNA, waiting for the right environmental conditions to activate, or whether they were introduced recently. Sarah returned to her desk and picked up one of Elena's classified papers.
According to this research, the sequences don't match normal human evolutionary patterns.
The phylogenetic analysis suggests they were inserted into the human genome, not evolved naturally.
But, inserted by what? Marcus asked. And, when? Sarah shook her head. That's the question that Elena's team has been unable to answer.
The genetic evidence suggests the insertion was recent, possibly within the last few hundred years, but the technology required to perform such modifications shouldn't have existed until very recently. Marcus was quiet for a moment, then looked up with an expression that Sarah had learned to associate with breakthrough insights.
Professor, what if the insertion wasn't recent? What if these sequences have been dormant in human DNA for much longer, and they're only now being activated by environmental conditions that didn't exist before? You mean like a genetic time bomb that was set to activate when humans reached a certain level of technological development? Sarah asked.
Exactly, Marcus replied. Think about it: electromagnetic radiation, chemical pollutants, artificial light exposure, processed foods, stress from modern life. All of these environmental factors are relatively recent in human history. Sarah felt a new wave of unease as she considered Marcus's theory. If that's true, then whoever or whatever inserted these sequences into human DNA knew that humans would eventually develop technology that would trigger their activation.
Professor, there's another possibility, Marcus said carefully. What if these sequences are activating now because they're supposed to? What if they're not random mutations or foreign insertions, but part of a natural adaptation process that helps humans evolve to survive in the modern world? Sarah considered this possibility, weighing it against the evidence of medical problems and neurological stress that seemed to accompany the activations. Marcus, if these sequences are meant to help humans adapt, why are they causing so many health problems? Maybe because evolution is never a smooth process, Marcus suggested. Natural selection involves a lot of trial and error, and not every genetic variation is beneficial for every individual. But, if enough people can successfully adapt to these new proteins, it could represent a significant evolutionary advantage for the species as a whole. Sarah walked back to her window, watching students cross the campus quad below. She found herself wondering how many of them might be expressors, unknowingly producing mysterious proteins that could either enhance their abilities or cause serious health problems. Marcus, we need to make a decision about how to proceed with this research, Sarah said. Elena has essentially invited us to join an unofficial investigation into one of the most significant discoveries in human history. But, doing so could put us in conflict with official government research efforts and potentially expose us to serious risks. Marcus stood up and joined her at the window. Professor, with respect, I don't think we have a choice. If these genetic activations are accelerating and affecting more people, someone needs to be working on understanding what's happening and how to help people cope with the changes. Even if it means challenging some of the most fundamental assumptions about human evolution and genetics? Sarah asked. Especially if it means that, Marcus replied. Professor, if humans are experiencing genetic changes that could reshape our species, don't we have an obligation to understand those changes and help guide them in a positive direction? Sarah turned back to her desk and gathered Elena's papers into a neat stack. Marcus, if we do this, it will change everything about your academic career. You'll be working on research that may never be publishable, investigating questions that some people don't want answered, and potentially discovering truths that could be dangerous to public safety. Marcus smiled, and Sarah recognized the expression of determined curiosity that had made him such an exceptional student. Professor, when I asked that question about the mysterious 8% in your genetics class, I wasn't looking for easy answers. I was looking for the truth, whatever that might be. Sarah made her decision. All right, Marcus. Let's find out what that 8% of human DNA is really doing, and what it means for the future of our species. She picked up her phone and scrolled to Elena's contact information. I'm going to tell Elena that we want to join Project Helix. But, Marcus, once we cross this line, there's no going back to simple academic genetics research. Professor, Marcus replied, I don't think we were ever doing simple academic genetics research. We were just pretending that we were. 6 months after Marcus first asked his question about the mysterious 8% of human DNA, Sarah found herself standing in a secure laboratory facility outside Washington, D.C., watching him analyze genetic samples from the largest study of human genetic activations ever conducted. Project Helix had evolved far beyond Elena's original research team. What had begun as a small investigation into uncharacterized DNA sequences had grown into an international collaboration involving geneticists, neurologists, immunologists, and researchers from fields that Sarah had never imagined would be relevant to human genetics. Professor Chen, you need to see this, Marcus called from his workstation, his voice carrying the same excitement that had characterized his breakthrough discoveries throughout their months of collaboration. Sarah walked over to Marcus's station, where multiple computer monitors displayed complex genetic analyses and data visualizations. Over the past 6 months, Marcus had developed analytical techniques that had revolutionized their understanding of genetic activations, earning him recognition as one of the project's most valuable researchers despite being its youngest team member. What have you found? Sarah asked, noting the unusual expression on Marcus's face that she had learned to associate with discoveries that challenged their existing assumptions. Marcus pointed to a genetic sequence displayed on his primary monitor. Professor, remember how we've been trying to understand why some people experience beneficial effects from genetic activations while others experience medical problems? Sarah nodded.
One of the most frustrating aspects of their research had been the apparent randomness of activation outcomes. Some expressors reported enhanced cognitive abilities and improved physical health, while others experienced neurological disorders and immune system problems.
I think I've found the answer, Marcus continued. It's not random at all. The outcomes depend on complementary genetic modifications that most people don't have yet.
He pulled up a comparative analysis showing genetic sequences from two groups of expressors. The people who experience beneficial effects from activations have additional modifications in their regulatory genes that help their cells properly process the new proteins. The people who experience medical problems are producing the proteins but can't regulate them effectively.
Sarah studied the data, recognizing the implications immediately.
Marcus, are you telling me that these genetic activations require multiple coordinated changes to be beneficial, and we're currently in a transition period where people have some but not all of the necessary modifications?
That's exactly what I'm saying, Marcus confirmed.
And, Professor, here's the really interesting part. The people with complete modification sets are passing them on to their children at much higher rates than normal Mendelian inheritance would predict. Sarah felt the familiar mixture of excitement and apprehension that had characterized their entire involvement in Project Helix. Marcus, that sounds like these genetic modifications might be self-propagating once they reach a certain threshold in the population. Marcus nodded and opened another file containing population genetics models. Professor, based on our current data, I estimate that within 2 generations, most of the human population will have either complete modification sets or no active sequences at all.
We're looking at a potential split in human genetics.
Before Sarah could respond to Marcus's statement, Elena approached their workstation carrying a tablet and wearing the kind of serious expression that Sarah had learned meant significant developments in the project.
Sarah, Marcus, I need to speak with both of you privately, Elena said. We've received some new information that changes our understanding of the timeline for these genetic activations.
They followed Elena to a secure conference room where several other senior researchers were already assembled. Sarah recognized Dr. James Wright from the CDC, Dr. Maria Santos from the World Health Organization, and Dr. David Kim from the National Security Agency's Biological Research Division.
Thank you for joining us, Elena began as they took their seats.
Marcus, your recent analysis of modification patterns has been extremely valuable, but it's also highlighted some urgent concerns that we need to address.
Dr. Wright leaned forward, his expression grave.
Marcus, your population genetics models suggest that we're approaching a critical threshold in human genetic evolution. According to your projections, the next 5 years will determine whether these modifications become established in the human genome or whether they cause widespread medical crises.
Marcus glanced at Sarah before responding. Dr. Wright, that's correct based on our current data. The activation rates are accelerating, and the environmental triggers continue to increase.
If people don't develop the complete modification sets needed to process these new proteins safely, we could see significant public health emergencies. Dr. Santos opened her tablet and displayed a world map showing activation hotspots. We're already seeing increased medical emergencies in several regions. Emergency rooms in major cities are reporting unusual neurological symptoms, immune system disorders, and sensory processing problems at rates that correlate directly with your identified activation zones.
Sarah felt a growing sense of urgency as she studied the global data. Dr. Santos, how widespread are these medical emergencies? Are we talking about isolated cases or epidemic-level problems? We're approaching epidemic levels in some regions, Dr. Santos replied.
But, here's what's particularly concerning, the medical problems are occurring primarily in adult populations.
Children born to expressor parents seem to be developing the complete modification sets naturally.
Dr. Kim spoke for the first time, his voice carrying the authority of someone accustomed to dealing with national security threats. Dr. Chen, Marcus, we need to understand whether these genetic modifications represent an external threat to human security or a natural evolutionary process. The distinction has significant implications for our response.
Marcus looked at Sarah, then back at the assembled officials. Dr.
Kim, based on our research, I believe these modifications represent an accelerated evolutionary adaptation to modern environmental conditions. But, I also believe the adaptation process was initiated by external intervention. The room grew quiet as the implications of Marcus's statement settled. Sarah realized that her brilliant student had just voiced the conclusion that all of their research had been pointing toward, but which none of them had been willing to state directly. Elena leaned forward. Marcus, when you say external intervention, are you suggesting that these genetic modifications were deliberately introduced into the human genome by a non-human intelligence? I'm suggesting that the genetic modifications show clear evidence of intelligent design, that they appear to be specifically adapted to help humans survive in technologically advanced environments, and that they're activating according to a timeline that correlates with human technological development, Marcus replied carefully. Dr. Wright consulted his notes. Marcus, if your hypothesis is correct, then, whoever or whatever introduced these modifications anticipated that humans would develop electromagnetic technology, chemical industries, and artificial environments that would require genetic adaptation. Marcus nodded. Dr. Wright, the modifications appear to provide enhanced resistance to electromagnetic radiation, improved processing of synthetic chemicals, better adaptation to artificial light cycles, and enhanced cognitive abilities that would be beneficial in information-rich environments. Sarah found herself thinking about the broader implications of what Marcus was suggesting. If these modifications were introduced to help humans adapt to modern technology, then, the intelligence responsible must have had detailed knowledge of human development and environmental needs. Dr. Kim's expression had grown increasingly serious throughout the conversation. Dr. Chen, the possibility that non-human intelligence has been modifying human genetics raises questions that extend far beyond public health concerns. Elena stood up and walked to the conference room's large display screen. Everyone, we need to discuss the immediate practical implications of our research. Marcus, based on your models, what do we need to do to help people successfully adapt to these genetic activations? Marcus consulted his laptop before responding. Dr. Vasquez, we need to identify people who are beginning to experience activations and provide them with medical support to manage the transition. We also need to understand and possibly replicate the environmental conditions that promote beneficial modifications rather than harmful ones. What kind of environmental conditions? Dr. Santos asked. Controlled electromagnetic exposure, specific nutritional supplements, stress reduction techniques, and possibly targeted therapies that help regulate protein expression, Marcus explained. The people who successfully adapt seem to benefit from structured support during the activation process. Dr. Wright made notes as Marcus spoke. Marcus, are you suggesting that we could develop protocols to help people safely undergo these genetic activations? I'm suggesting that we should try, Marcus replied. Because the alternative might be watching a significant portion of the human population suffer medical crises as these modifications continue to activate without proper support. Sarah looked around the conference room at the assembled researchers and officials. 6 months ago, she had been teaching basic human genetics to graduate students. Now she was participating in discussions about humanity's genetic future and the possibility of non-human intervention in human evolution. Elena returned to her seat and addressed the group. We need to make some decisions about how to proceed. We can continue treating this as a classified research project, or we can begin developing public health protocols to help people manage genetic activations. Dr. Kim shook his head. Dr. Vasquez, the national security implications of announcing that human DNA has been modified by external intelligence would be catastrophic. We're not prepared for the public panic that would result from such a disclosure. Marcus raised his hand hesitantly. Dr. Kim, with respect, if we don't help people understand what's happening to them, the medical crises and social disruption might be even more catastrophic. Sarah felt proud of Marcus for speaking up to the NSA official, but she also recognized the validity of Dr. Kim's concerns about public reaction. There might be a middle approach, Sarah suggested. We could develop public health protocols that help people manage unusual symptoms and genetic changes without necessarily explaining the full scope of what we've discovered. Dr. Santos nodded thoughtfully. Dr. Chen, that's similar to how we handle many public health challenges. We provide practical support and medical guidance while continuing research into underlying causes. Elena looked around the conference room.
Are we in agreement that we should proceed with developing support protocols for people experiencing genetic activations, while continuing our research into the origins and implications of these modifications? The group reached a consensus to move forward with practical applications of their research while maintaining security around its most sensitive discoveries. As the meeting concluded, Sarah and Marcus found themselves walking back to their workstation together, both lost in thought about the magnitude of what they had uncovered. "Professor," Marcus said as they approached their laboratory area, "when I first asked you about that mysterious 8% of human DNA, I thought I was just curious about gaps in scientific knowledge." Sarah smiled at her former student, who had become one of the most important researchers in human genetics. "Marcus, your curiosity about those gaps led us to discover that humanity might be in the middle of the most significant evolutionary transition in our species' history. And we still don't know if that transition will ultimately benefit us or threaten our survival," Marcus added. Sarah looked out through the laboratory windows at the world beyond, knowing that millions of people were going about their daily lives unaware that their genetic code was quietly changing. "Marcus, I suppose that depends on how well we help each other adapt to whatever we're becoming." As they returned to their research, Sarah reflected on the journey from Marcus's simple classroom question to their current position at the forefront of humanity's genetic future. The mysterious 8% of human DNA had revealed itself to be far more than junk sequences or evolutionary remnants. It was a genetic roadmap for humanity's next evolutionary step, and their research would help determine whether that step led to enhancement or catastrophe. The question that had started it all, about uncharacterized sequences in human DNA, had opened a door to understanding that humanity was not just evolving naturally, but was being guided through a carefully orchestrated genetic transformation whose ultimate purpose remained a mystery. And, somewhere in the background of their ongoing research was the persistent question of who or what had initiated that transformation, and whether their intentions for humanity's future aligned with humanity's own hopes for survival and prosperity. As Sarah dialed Elena's number, she realized that Marcus was right. The moment he had asked about that mysterious 8%, they had crossed into territory that would challenge everything they thought they knew about human biology and evolution. The question now was whether their discoveries would ultimately benefit humanity or threaten its survival.